Okay, let us begin. So I'm not sure how to construct it, but I'll construct it this way and kind of jump around in an effort to get most of the material done for the rest of this first chapter. But probably won't get all of it because we're not here Monday, of course. So if I miss something, if I feel like I want to get started in the next chapter, I'll probably send you something. I'm having trouble at home doing that because I keep forgetting to turn on the recording. So I did that yesterday with another class. I did a whole lecture, and I said, "Oh, I forgot to make it record." So I spent it. The dog's looking at me, going, "What are you talking about?" At least he's funny. He's one of those. He looks, and when he's looking at you, he gives you one of these. All so this kind of comes toward. It's sort of intermixed in this chapter. It's the concept of what we're really talking about. So when we're talking about microbes, it's things we can't see, but we have elements that are living and non-living. So I figured I'd do this part now because I usually do it on the board, and I like to do it in person. Rather than have to do it something virtually, because it's a little bit easier, I think, for me to demonstrate. Living things, like us, we're all made out of cells. By that, living things by definition, not us. We're obviously macroscopic. You can see us. Made out of cells, so we are multicellular by definition. Understand in the microbial world, most things are single cellular if they're celled at all. Okay, so bacteria and viruses, which aren't living, we'll get to that. But bacteria, by definition, single celled, so obviously you couldn't see them with the naked eye, so they have to be microbes. Cell wall or not. So we don't have cell walls in our cells. You all know that. Plants do. Big distinction, and that's why, at least historically speaking, everybody kind of thought that the bacteria were somewhere in the plant family. And I mentioned this before. You hear the term microflora or normal flora a lot, particularly for those who endeavor to work in a hospital or a medical environment. So by definition, living things don't have to have a cell wall like us, or they can. They obviously have to have boundaries are important because boundary isolates you as an organism. If there's no plasma membrane, not a living thing, because it controls the entry and exit ways. So for those who had me, and a good many students had me for A&P, I went slightly nuts over movement across membranes, osmosis, facilitated diffusion, simple diffusion, all that type of stuff. There's a reason for that, because really that kind of controls our environment, what's going in and what's going out. All of that's just so terribly important. So the boundary aspect of the plasma membrane, the fact that basically our interactions with the environment, particularly on a microbial level, come from things that attach that plasma membrane. More on that down the road. Living things have DNA and RNA. There's no or. It's an and. Why? The DNA facilitates replication, making more of them, because you have to be able to reproduce. And the DNA functions for transcription. And just remember, that's basic. What's transcription? DNA making RNA for the purpose of making proteins. Because we get the simplest thing about genetics that we all rely on is DNA making RNA and RNA making proteins, and that's what genetics are, what our proteins say we are. That's a distinctiveness of us or any other organism. So what's RNA's job in this grand scheme of things? Basically making proteins. We call that translation, translating what DNA, the language of DNA, and making it effectively into proteins by way of a series of RNA-based translations. The ability to respond, this is, the responding to environment, okay? So we have things like, people, they talk about human beings, you have to have a nervous system and changes. All living things do that. They just do it differently. When we see with bacteria, as the environment gets warmer, they do something. If it's colder, they do something else. If there's certain chemicals in the environment, they do something else, right? They may be attracted or repelled to them. It's very simplistic. It's not that they have to make dramatic adaptations. Microorganisms are generally extraordinarily simple. So they just respond to what's there. if you put something in there, by and large, they look at it as a nutrient and they gobble it up. That's one of the great things about things. We talk about chemotherapy, and I make this distinction now. When you hear the word chemotherapy, it doesn't implicate that is a cancer treatment. That means treating someone with a chemical. You're taking an antibiotic as a form of chemotherapy. Okay. Me taking a blood pressure pill in the morning, which I do, is a form of chemotherapy. It doesn't mean so. I mean, names, words matter in that regard. Okay. So bacteria, we throw something in their environment. First thing it looks like, it's a nutrient. It's like the dog. You put something. It's food until otherwise stated. So they respond differently to things in the environment. How you'll see things attached to different receptors on their membrane, and they respond to it by either moving towards it or moving away from it. And then what all living things have to do, so reproduction, and you're going to see reproduction is very simple when we start looking at bacteria. Okay, it's very different when we're looking at cells like ours. Okay, but there has to be some ability to reproduce. And lastly, and perhaps as important as anything, our metabolism. Effectively, the way I sort of look at it is what's called energy transformation. In some way, shape, or form, microorganisms have to be able to obtain nutrients from their environment, break them down, utilize them for energy, and get rid of waste products. Just like you and I, we eat something, we utilize it for energy, we get rid of the waste. Do we use some of it as building blocks? Sure. And they're no different. Living things all do that, just in a different manner. Some is more complicated and some is more simple. Obviously, all about microorganisms simple, but that's the basic sort of job description and basic materials that you need to have to be considered a living thing. So over here, this is for prokaryotes. You might remember we talked a little bit about the name pro before it had the pro or in development or not visible, the carrion, which is this core or central area, and what scientists basically thought that as the nucleus that's here. So these are non-nucleated cells, but a better way to describe them, no membrane organelles. So prokaryotes, and this is where we're getting into the nomenclature of it. This is the domain system. For years, again, bacteria and things like that were offshoots of the plant kingdom. Then in 1978, Weiss, and I think you'll see the citation here, and I'm not quite sure if that's the pronunciation, Woese, and I'm probably German, but I don't know it. So that particular scientist was studied in the age of molecular biology. He and his associates began to study sequences of RNA and DNA because they could do so in the realm of molecular biology. You could see the components. And they developed a classification system based on change within the DNA, and interestingly, the density of the RNA, because now you're going to have to know about this bad boy, the S unit. S stands for another scientist whose name is S units. I can do the eukaryotic things over here. I'm so happy. Svedberg unit. And they're measures of density. I wouldn't know a Svedberg unit if I fell on it. They were counting Svedberg units in my backyard. I closed the blinds. I have no interest. It's kind of like watching, Miami going up to playing Buffalo. Wouldn't watch it in my backyard. Okay. So, prokaryotes. He, based on the densities and the structure of the DNA, created a system of 3 domains. 2 of those domains were considered prokaryotic domains. Why? Because they contained very, very simple forms. They were single cells. They all had a cell wall. They had regular DNA. That's the beauty of DNA. The DS stands for double-stranded. Our DNA your cells, my cells, bacteria cells, all the same. Double-stranded DNA, the old double helix thing, they studied, they have it on the PowerPoint here, over it. Not that interesting. Okay, twisted ladder, the whole deal, A's and T's, G's and C's, that whole thing. Good. So when you see double-stranded DNA, when we get to viruses, they actually have, go figure, some single-stranded DNA that we don't really get into the screen. We start to, just to me, that's it. More information. Our DNA, so if we're in a eukaryotic cell, this gives me the opportunity to do this. So in eukaryote, that's us. You with the carry-on, the nucleus, we have, okay, totally, it's quite a bit different. So if you look at it from that construct up there, it can be single or multicellular. So you're going to see eukaryotic organisms. You can see eukaryotic organisms that have one cell. I even talked about that with that Trichomonas vaginalis that causes the second most common sexual transmission. One cell. Or they can be multicellular, like us. But they're us, we're too big, we're not like us. So eukaryote, single or multicellular. Okay? They can have a cell wall or no cell wall. So there's a certain amount of freedom to it. We'll get into that. They have DNA, of course, double-stranded. I'll put that DS. But the DNA, and you all know this, is in pairs. Linear pairs. We've all seen it. 23 pairs of chromosomes. It's arranged linearly. That's a very significant difference, particularly when we get into the genetic aspect. Because it's one of the things that protects you and I from mutations. Bacteria don't have that protection. Because they're pairs. What does it mean to have a pair of DNA? And you all remember basic genetics, dominant and recessive. That way, if there's a mutation, and occurs on, and if there's one dominant and one recessive, and let's say if the mutation, the error occurs on a recessive strand, it doesn't get shown up, dominant shows up. So you're inherently protected. It's a really important concept. What do we call organisms like you and I, or eukaryotic cells with pairs of DNA? A great word for them, you already know it. They're called diploid. Remember that? That ringing a bell? Can't hear you now. Yes, right. We all remember diploid. So what does that mean with this? This is like a sperm or an egg. Okay? It's got a single piece of DNA. So by definition, this is haploid. That means it's much more subject to genetic. Back to genetic. Good primer. So you have DNA. The RNA in eukaryotic cells, it's confusing. ADS. Just wait till we get and look at the organelles. Get more. So what does that mean? The difference between 70S RNA and 80S RNA is just denser. I want to think of that as heavier. So more, units. Remember, DNA, RNA has a smaller subunit and a large subunit. When you pack them together, it's a little, based on this, I guess, a little heavier, denser, however they do. Okay. And what does it have an abundance of? Complexity. Read that. It's membrane-bound organelles. How does it reproduce? Mitosis and, or, because not everyone does it, meiosis. So it reproduces, all that stuff, prophase, metaphase, anaphase, telophase, cytokinesis. Mitosis or meiosis, just like us. These guys, much more simple. They just divide in two with some very rare exception. Called binary fission. Binary for two, they just divide in two. They do energy transformation, sure. Made out of cells? Sure. Those are your differences. So where are your big differences between prokaryotic and eukaryotic cells? We can look at the structure of the DNA. We can look at the 70S versus the 80S. We can look at the cell wall, which they have, which these don't necessarily have. And what those differences are, when we begin to look at it in a larger sense, particularly in a disease-oriented sense, those are areas that. So therapeutically, if you get sick with a microorganism, so let's say what's as common an infection as there is with bacteria, which is in the prokaryotic realm. Bacteria live everywhere. They have cell walls, and those cell walls contain very interesting chemical. It's a building block called peptidoglycan. Peptido means chains of amino acid, peptide bonds. Remember those from AP1? Bond, peptide bond. See, you guys. And the first James Bond movie came out 60 years ago. I was. Late Sean Connery, 1962, Dr. Young. I had been reading the books before. A very interesting author named Ian Fleming, who you see a lot in World War II spy movies because he was really part of the SOE, the Special Operations Executive. It was sort of like the dirty tricks unit, sort of along the lines with our OSS was the case. A little history doesn't hurt. Peptide bond. So it has amino acids and peptide bonds, and glycan means sugar with nitrogen. So you're going to see this building block. Don't have to worry about it now, just know the name, because all bacteria have it. Because all bacteria have it, it's a target. So if we can get something that attacks peptidoglycan, we can attack bacteria. So if we play word association and you have an infection, what do you take for a bacterial infection? What do they prescribe? You get a prescription for a blank. Sir, you get a prescription for an antibiotic. And you play word association. See, it's not that hard for us to all participate with one another. Now, if I say antibiotic, what's the first name that comes to your mind? Go ahead. Penicillin, God bless you. Okay, what does penicillin target? Peptidoglycan. What are the best categories of antibiotics? Penicillins. Because they attack peptidoglycan, and guess what? Only bacteria have peptidoglycan, we don't. That's a concept called selective toxicity. That's the foundation of therapeutics. It'll kill the bacteria, it shouldn't affect us. But what's the problem with antibiotics? With penicillin? What do people have that doesn't allow them to take? Surely someone in this room has a problem taking penicillin. Sir, they're allergic. Look. I was saying that in a global sense, not a specific sense, because you get into trouble. See, that was good. So, what do you take something when you're allergic to penicillin? You don't know it yet, but I'll tell you. Basically, the drug of choice now has become the Z. You're going to learn about this first. Cancer is zithromycin. Zithromycin is in the family of, it's got a fancy name you're going to learn called macrolides. And it's called erythromycin, is the original compound. What does it target? Another difference. It targets 70S ribosome. And the difference is, the big problem we have with that is not so much allergies, but side effects. Because even though eukaryotic organisms have an 80S ribosome inside some of their organelles, for us, particularly the mitochondria, and we'll get to it, we have some 70S. So even more so, now bacteria are everywhere. The difference between archaea, and I said this before, they're older, they're in more extreme environments. Think about like when the planet was born or in the ice age, that type of stuff. They don't have any bacterioglycan. Thankfully, at this point in time, we do not think that they impact us. And I don't spend a lot of time talking about it. They're more in research, almost in a sci-fi type of sense. About this stuff. Okay? So, the techno thrillers and things that we talk more about, because they're very, very interesting. They're probably a way of the future. Look at them as we go to extreme environments. Interestingly and oddly enough, and it's not an important point, but for the sake of completeness, our DNA is more similar to that DNA. I don't know what that means necessarily. That's something scientists will probably. So those are the categories. Now the domain of eukarya, our domain, has microorganisms in it. They really fall into two major categories. Algae, actually three if you want to think of it. Okay. But algae, what makes algae unique? Again, the word unique is very important. Unique doesn't mean, Patrick Mahomes is a unique quarterback. He's a great tremendous skill set. But it's not a one of a kind thing your other quarterbacks do. Unique in science means no one else. So peptidoglycan is unique to bacteria. What are unique to the microorganisms in the eukaryotic domain are algae is that they contain, chlorophyll, which means they are photosynthetic. That's that whole nugget about being able to produce, according to this text, about 70% of our oxygen from all that algae that are in all the seas. So the only photosynthetic microbes that are impactful to us and the only are algae, and algae are the only ones. The others, sort of in the realm of what are called protists or protists. Okay, and algae are in that they're protists. Algae are like plants; they have a cell wall. They're eukaryote, they have a nucleus and all of that. They can be single or multicellular. Single. And basically, what's neat about them is the photosynthetic. So it's like they gather their own energy and food sources much more. Another protist, or something that we see particularly, and these are water. The other ones are called protozoans. So when you were in high school and you looked at pond water, most people did. Those neat little things that were swimming around and spinning around. Over there. I was called swimmers and spinners. Spinners are very good old motel. This shows you how long ago it was. They did a song called The Rubber Band Man, which was the theme song for the Fast Times at Pirates. Because their reliever, Kent Colvin, was over her. God, it's so that's what we practice. So, protozoans, all right, say things, we look at them a little bit at these called rotifers, which do the spinning and amoeboid motion, and flagellants like that trichomonas vaginalis, they all move, their movements are exactly. But protozoans are like that, but interestingly, they are more like us. They're a single cell eukaryotic cell. No cell wall, single cell, not photosynthetic. And you see them typically in soil or water. So that's another one. So you have the two protist categories, if you will, with the difference in the algae having being mostly the photosynthetic nature of the chlorophyll, and the fact that it can see cellular. And then lastly, of the significant ones are these, and it's not to underscore protozoans, like I told you, malaria is a protozoan. It comes from that. And that's a monstrous disease on a worldwide basis and a monstrous killer. And then lastly, this, which is another one of these singular multicellulars, these are yeast or fungi that are there, mold, if you will, is in that category as well. They can be single or multi-cell. They have a cell wall, so they're more like plants, but they don't have any chlorophyll. They're more like us. So they're sort of a hybrid. And what's very interesting about them is they play a really significant role in importance because most of our antibiotics come from the aforementioned penicillin. Maybe the best antibiotic around, which is a cousin of that, called cephalosporin, that we'll get to. And these have no chlorophyll. And they're interesting. They're what we call decomposers. Their fancy name in the microbiology, they're called saprophytes. And that means they gobble up dead stuff. They're the roadkill cafes. So bacteria and fungi and mold are like that. Get all that in. Less than a half an hour. Nicole, I'll take a picture of that and send it to you. I have on the board. Jeez. Not little kill, roadkill. I like to be correct. So that kind of, on our outline, that kind of took us on page 2 of the outline. The domains, archaea, bacteria, eukarya, etc. The nomenclature, I just want to get through this. The nomenclature nugget is genus and species. I talked about it before. So we don't have to go into all the orders and families and stuff in traditional biology that are there. What I like is that it's very simple. All you have to do is use the first name, and the naming convention has a lot of different. Like, for instance, most people hear about, MRSA. Well, MRSA means methicillin-resistant Staphylococcus aureus. The bacteria, methicillin-resistant means it's resistant to a particular category of antibiotics, which are in the penicillin family. We do a whole thing on that. Staphylococcus is its name. It's not named for a person or a location. You're going to see it's named for its structure, and we get to that relatively soon. Staphylo means a cluster, and cocci are little spherical subunits. So it's like a bunch of grapes, really what it looks like. Okay, microscopically. And aureus, the AU, periodic table, meaning it has a golden color. The colonies, and sometimes the pus it produces is golden in color. See? You got to use the periodic table. That's another James Bond thing. A very famous Goldfinger, his first name was Auric, not Eric, A-U, who's named for gold. Oh, Lord help me. I'm a fountain of useless information. But again, a great partner for Jeopardy if it was questions before 1980. Guaranteed winner. And my mom still got the answers before me when she was in her late 80s. So here we are, prokaryotes, eukaryotes, the domains, we got all that. I'm just trying to make sure I've got everything covered before I go back and sort of scroll through the PowerPoints. Study rapid reproduction. And that's the beauty of why, again, we use them so much in research, because thankfully it saves us from experimenting on animals extensively, and we did so much of that historically. It doesn't exclude it, but it does save a great deal of it. And the rapid reproduction, as you'll see, it's really extraordinarily important because they replicate so rapidly. The medical microbiology is coming, and particularly I wanted to talk a little bit more about genetic engineering and gene therapy that are there. So that was in that particular nugget. These additional notes are kind of what I put on the board. And really, we extensively, maybe the linchpin of the chapter that we're coming to is that peptidoglycan in the cell wall. And really, in the chapter where we start looking at this, which is, I think, chapter 3, just like it says here, is in looking at the gram negative and gram positive structure. Because by this time, you all have microlab, I'm guessing, by now. Okay. So basic microscopy is what you did by and large. Did you do any staining yet? Not yet. You're very fortunate. I mean, the instructors are great. They're two of my favorite colleagues. Between Dr. Dress and Dr. Hill, we'll have a great time. So let me see if there's anything else that's there. No, oh, that's the domain thing. And we did the historical figure. Here's the additional note. Genetic engineering. Bacteria, and I'm a horrible artist, and we have wonderful animations in these chapters that will demonstrate this to you. How can I say it? In bacteria are very interesting, and it goes back to the genetic aspect of it. They have the remarkable ability of being able to change genetically in the same generation. Literally, a person with a staphylococcal infection in the presence of one MRSA bacteria, theoretically in that population, can change it overnight to all MRSA. It's like you and I. If we were in a class and we had one great athlete, and that it's almost as though that one athlete could touch the person next to them, and they could touch the person next to them, and so on throughout the whole class, and all of a sudden, magically, we'd all be great athletes. We'd all be Michael Jordan. We'd all be LeBron James. We'd all be, please be Tiger Woods for a day, just like a big golf ball like that. That we could do that. Bacteria can do that because of that business genetically, that simplistic nature. Bacteria, you and I don't have these. There are some eukaryotic cells like yeast that are very, very simple, can have these. They're called plasmids. That's the name that's there. Okay, so we utilize both E. coli, which we have a lot of, and the E. coli we use is this very, very special strain that's not harmful, but genetically very malleable. We can play with it genetically. DNA is resequenced with plasmids. Bacteria have these tiny circles of DNA. Okay, in addition to their own big circle of DNA, most of them have these plasmids, which you're going to learn are non-essential, meaning they can get along with them or without them, but most of them have a bunch of them. And those plasmids can move from one bacteria to another by a magical process called conjugation. And we utilize this as part of how we do genetic engineering. We attach, because we can make our own DNA, laboratory-made sequences of DNA, splice them, okay, just attach them in the laboratory to the DNA of a plasmid and move that plasmid from point A to point B. Read that as bacteria A to bacteria B. And effectively, that's what genetically modified organisms do. We can resequence them, and then, because bacteria reproduce, effectively, that protein, insulin, is a waste product that we can collect in the laboratory. It's hepatitis B vaccine. It's human growth hormone. It's Pitocin. We can do all that. So the DNA manufactured in the lab, splice it, insert it into E. coli or yeast, and we can make all those things with it. That's the beauty of molecular engineering. There's good and bad. I mean, whether it's philosophical or ethical, modifying things by direction, is something that people can be troubled about. And you read a lot of that. I mean, and there's a whole lot of those things, whether it's organic gardening that we do, or whether it's making people not wanting to consume genetically modified organisms for whatever reason that are there. That's fine. Yeah, I think you have to suit yourself, but some things are certainly of value. And gene therapy, and I commented before, we utilize the HIV virus because it contains an enzyme, and we can restructure what the information of the virus and effectively use that to insert it in certain kinds of cells particularly white blood cells, and resequence them. Okay? We have two major categories of diseases, emergent and resurgent. What's meant by that? Emergent diseases, okay, new diseases, either, as I've described it here, either recently discovered or as the result of genetic mutations that appear and cause problems. COVID is clearly a disease that's emergent. Now, again, you can get into all sorts of chapter and verse of how, whether it was emergent by design or emergent by accident, not a discussion for this class. Okay, because there's too much politicizing of the disease, in my opinion. But effectively, what happened, most of the problems we have, MRSA, okay, BRE, aggressive strains, killer strains of the flu, killer strains of COVID, have to do with mutations. Okay? Again, bacteria and viruses mutate relatively easily comparatively. They just do. They don't have the genetic protection and the safeguards that you and I have, and we go into this in more detail in the chapter on genetics. The flip side of that, okay, is a little bit different, and that's the so-called recent discovery. We're going into areas, so when we go in, as the population expands and goes into areas like rainforests, we begin to encounter different kinds of problems. When we have a larger population in an area that was not as much inhabited, we have a greater amount of problems. So if you recall back it was in 2014, in that time, you had two major events in Brazil, and a lot of the venues were sort of carved out in the rainforest. They had the World Cup and they had the Olympics. Okay, and we all of a sudden magically start hearing about a virus called Zika, which is a virus that's in the same category with, interestingly, as hepatitis C and yellow fever, but still it was new in the sense. So did it just magically form, or is it because we went into an environment that was new and it came upon us? That's possible. So emergent diseases, my way of thinking, are mostly mutation, but a lot of times it's population or living space expansion, if you want to look at it that way. Resurgent diseases are diseases that were near eradication and are reappearing in larger numbers. And then I give you the reasons behind it. And a lot of them are sort of intertwined. Surely the two poster children for this are tuberculosis and measles. We'll start with TB. TB has one basic source of problems: overcrowding. You have to be exposed to tuberculosis for long periods of time for it, because it grows very slowly and it's comparatively difficult to spread unless you're continuously exposed. That's where overcrowding. So it had to do why it was so everywhere. If you read back, if you watch historical novels or historical fiction or movies, from period times, they're constantly talking about a disease called consumption. That's tuberculosis. Okay? And what it means is the organs are being consumed within. Tuberculosis, we go over, it's a respiratory disease that can spread through all your body. So it's a bacterial disease that's kind of like cancer. Very interesting. We nearly eradicated it in this country without a vaccine, through screening and treating when necessary. TB tests, which everybody I'm sure has had one, chest x-rays, identification, blood tests to see if there was a problem, and then treatment if necessary. And it was nearly eradicated. And then all of a sudden, we began to see a remarkable uptick in it, and it tended to coincide with the age of immunological disorders, particularly HIV. And what's called an AIDS-defining diagnosis, and there's many, is someone who's HIV positive and gets a particular disease, and all of a sudden they make the transition to AIDS. They go from being treatable to being fatal. TB is one of those. So if an HIV positive person gets TB, now they got AIDS, period. They're going to die sooner than later. That's what fatal means. Not going to live their expected lifespan. We nearly had it eradicated. Is that pointing a finger solely at that? No, not necessarily. Interestingly, there isn't a particularly good immunization for that. If you travel to foreign countries, Some of them will require that if you go to India for any reason. And India, and it's because of the massive amount of population in the Indian subcontinent, you have to get an immunization with something. It's interesting. It's called BCG, and I'll give you the whole story on it down the road. When we look at it, it stands for three doctors: Basil, Calumet, Guerin, which you don't got to know. Okay, and they take it from a similar disease, kind of like cowpox. It's called Mycobacterium bovis. And what they did is they extracted that, and they were giving it, and it works a little bit better in children than in adults. But even as an adult, to get a visa to go to India, you have to have that. And the trouble why we didn't use it in this country is you get a false positive. From now on, the TB test doesn't work for you. You're always going to test positive because you get a lookalike reaction. Yeah, my daughter had that. My daughter, maybe I told the story in A&P, maybe I didn't. My daughter made and I'm not recommending this. Believe me, I am not. A lot of money being an egg donor. Okay. So she donated her eggs, and because it was for infertile couples, will pay anything. Take it from one. Okay. Okay. Good. I have three. If you did not know, I have three adopted children. It ain't cheap. Okay. So they would pay for girls that meet whatever their criteria. Read that as nice looking, okay, and physically healthy, and it is, okay. Egg donors, and it was cheaper to do the procedure in India, not necessarily less safe, and they pay to be there, pay somebody to go with you, all sorts of fertility drugs, and you're there for X amount of weeks, and they harvest the eggs, and if you're a repeat customer, if somebody was happy with the first, then they'll pay even more money for the second. I'm not recommending it. She did it a number of times. My daughter's very enterprising in that regard. It's okay. But I digress about that. But she had to have that immunization. I went through the whole thing, and it was very, very interesting. And she did some stateside as well. Nevertheless, the other one's measles. Now we're hearing a lot of, and the problem is with measles is the first vaccine laxity. There's a certain extent, it's called out of sight, out of mind. Okay? None of you, anybody here have measles? I don't think so. Any chickenpox people? Because of immunizations. German measles, mumps? No. Because MMR, measles, mumps, and rubella has been around the longest. All of my kids did not have the measles or the German measles or the mumps. They all had the chickenpox because the chickenpox vaccine didn't come out until 1990. And they all had it within 2 weeks of one another in 1988, in order of their ages. That's how contagious it is. When I was a kid growing up, every kid, okay, that I knew had measles, chickenpox, German measles, and many had the mumps. It's the only one I missed. Lucky me. You see, talk about child abuse. This is an absolutely true story. I love telling this story. Ruth, you did this, pointing at my mom, who passed away in 2011. Okay. God bless, 93 years old. They all, at the end of first grade, we were all in the neighborhood with a lot of kids because everybody had moved out of the city into the suburbs. He said, we're all going to the circus. It was a Ringling Brothers Barnum Bailey Circus in the old Madison Square Garden, not where the new one is, in New York City. Great day, 40-minute bus trip. We all ate at the Automat. It's a great series. There's a great little thing on HBO about the Automat, a little documentary. It's hysterical. It's got a lot of Mel Brooks in it. So I think we were having a great time. Within 2 weeks, everybody in the neighborhood had the measles, and then 2 weeks later, we all had the chickenpox. Those were called measles and chickenpox parties. The moms did this. If they did this today, they would be cited for child abuse. But that's what they did. They were unhappy if you didn't get a good dose of it. Oh, little Mikey's only a little bit sick. I'm disgusted. He hardly has a spot on him. Nowadays, and we'll go into it. There's been both information and some misinformation about vaccines. I have my own opinions about them. Uh, but measles, mumps, and rubella has been a very, very trustworthy, as is the chickenpox, a very good immunization. And I'll talk more about that. But people, and when we've seen outbreaks, they have typically been, and there was one at Disneyland, not Disney World, within the last 4 or 5 years with an unvaccinated population that had come from, because, and interesting, in California, the laws were fairly liberal, at least at that time. Worldwide travel. We're going all over. This was part of how the original SARS, which is the parent, the first time we saw and heard of deadly COVID viruses, occurred, because people were going from, I think it was Tokyo to Canada, on an airplane with an active disease in a closed environment. But this is one of the reasons where diseases can spread in any event, but also played a role in this. Aging population. Don't start. I know what you're saying. You're part of it. Okay. Aging population. As you live longer, diseases can come back and make a reappearance that maybe you were immunized for, and over time the immunization wears. That's there. Immune system diseases, which not just HIV, but a lot of immunological diseases are recognized. I have an immunological form of arthritis. I take injections for every other week. And that's part of that is immunological suppressant therapy. So there's a lot of little nuggets that are all part of this kind of enlarged scenario. So there's a lot to it microbiologically. And this is most of the material. I have a few little odds and ends, and I probably can do those Wednesday without having to do more about that. But I wanted to give you some ideas about what goes on. And it's really the impact they have. When we talk about industry and medicine, we're so reliant upon these microbes, and they can be used for good and for bad. Like the whole the vaccine thing, and some of the, and like I said, again, a lot of that, I think a lot of the laxity came from some faulty research that was done about 20 years ago. But the reality is the research was faulty, but the warning was true about some of the things, and we'll get to that, in germicides and disinfectants. It's the stuff they put in vaccines is preservatives. And some of them are toxins, and we give them to babies, significant. So, I mean, and there's a lot going on with regard to all of these scenarios. So I leave you with that. And I hope you have a great weekend. We'll see everybody on Wednesday. I will take pictures of this and put, and I guess I can pop these on Blackboard. If you want to take pictures, go ahead.